The overall goal of this procedure is to reproducibly prepare a tissue sample for peptide mass spectrometry imaging.
This method allows novice mass spectrometry users to develop their own imaging mass spectrometry workflows without going through lengthy empirical determinations. The main advantage of this technique is that it is highly reproducible, simple to implement, and uses cost-effective methodologies.
To prepare the nitrocellulose-coated slides, pipette 40 microliters of liquid nitrocellulose onto one edge of a conductive indium tin oxide microscope slide. Using a regular glass microscope slide, drag the nitrocellulose under surface tension across the slide to create an even thin film coating. Allow the nitrocellulose to dry at room temperature for 20 seconds and then store until needed at room temperature. Next, create custom vapor chambers by cutting a piece of thick blotting paper big enough to fit the bottom half of a standard plastic petri dish. Cut the paper to leave a rectangular strip in the center the same size as a microscope slide. Leave enough excess so that the paper will maintain its position in the petri dish. For FFPE tissue, float mount the sections onto the nitrocellulose pre-coated ITO slides as described in the text protocol. Submerge the slides in fresh xylene to remove residual paraffin for 2 minutes. Wash the deparaffinized samples by submerging the slides in a graded solvent series consisting of a 70% volume for volume ethanol water solution followed by 100% ethanol for 30 seconds. Then, submerge the slides into Carnoy's fluid for 2 minutes.